Before we begin our study of groundwater pollution and the challenges of prevention and cleanup, we are going to do an in-class investigation. As surface water travels downward through the ground, it is filtered. As a result, the water in aquifers tends to be cleaner than surface water. For this experiment, you will work in groups of 3 to 4 students. This experiment will demonstrate how the layers of the earth can act as a natural filtration system of surface water as it seeps through the ground. This video will provide instructions along with an example of the process you will complete in class. Remember, before you begin, to put on eye protection, lab aprons or lab coats, and to wear gloves throughout the experiment. First, you will need to collect your supplies. For the filtration setup, you will make a total of 5 filtration apparatuses. For each setup, you will need one 750 milliliter beaker or a wide mouth pint canning jar, an empty 2-liter clean pop bottle that has had the bottom cut off and holes drilled in the lid, 2 cups of gravel, 2 cups of sand, 2 cups of soil, and a 1 cup measuring cup. Next, you'll gather your supplies for the experiment. They are located on the teacher's lab bench. You will need 1 sample jar labeled dirty water, 1 sample jar labeled oily water, 2 sample jars containing an unknown sample. Make sure these are different from each other. You will need 1 empty jar that will be used for the control, a ruler, and test strips. You will also need a ring stand and ring. Before you begin the experiment, make sure your pre-lab questions are completed. The teacher will come around to each workstation to look at your pre-lab questions as you collect your materials. You will complete 5 trials, 1 control, 2 known samples, and 2 unknown samples. To create the filtration setup, you'll screw the cap tightly onto the 2-liter bottle. One student will hold the bottle cap down while the other students fill it with the filtration materials. First, add 2 cups of gravel, followed by 2 cups of sand and 2 cups of topsoil. The filtration setup should be placed on the ring stand above a collection jar or beaker. The bottle will need to be held to keep it stable while the samples flow through the filter. Record your observation of the sample prior to filtration on your data table. For the control and unknown samples, use test strips to test and record data about the sample's pH, glucose, and starch levels. For the oily water sample, use a ruler to measure the amount of oil in centimeters and record that on your data table. One student should place the filtration setup on the ring stand above the collection jar or beaker. The bottle will need to be held to make sure it is stable. Another student should pour the sample in the filtration setup. Record your observations of the filtrate in the appropriate location on your data table. For the control and unknown samples, use test strips to test and record the pH, glucose, and starch levels. For the oily water sample, remeasure the amount of oil collected in centimeters and record the data on your table. When you have filtered each sample and recorded all observations, use the following instructions located at the end of the procedure to clean up your workstation. Pour the liquid samples into the matching waste containers located in the fume hood. Wash all glassware and return it to the teacher's lab bench. Dispose of the filtration material in the waste container labeled filtration waste. Wash the empty pop bottles and lids and return those to the teacher's lab bench. Used test strips can be thrown away. Return any unused test strips to the appropriate location on the teacher's lab bench. Clean up the bench at your personal workstation. If there is time, return to your seat and work on the post-lab questions or complete them as homework.